last video, we talked about some genetic conditions that you can wind up with. Now we're going to talk about some issues with the actual chromosomes themselves. So this thing called polyploidy, I don't know why I highlighted this one, but not this one, but there's this thing called polyploidy, which is an aberration in the number of chromosome sets. Meaning, if you're polyploid, you have too many chromosomes, for instance. So lots of animals are diploid, which remember means they get one set of chromosomes from their mom and one set of chromosomes for their dad. And sometimes organisms are formed with, more than one, are formed with more than this diploid set. Those are called polyploid. So if you have, for instance, 3 chromosomes instead of 2, you are called polyploid. Polyploid plants are more robust, so lots of crops love to be polyploid. They don't mind it at all. Most of the time, polyploidy is lethal for human beings. Aneuploidy is a related but different term. This is an incorrect chromosome number. So members of the same species always have the same number of chromosomes. So you, your parents, your neighbors, you have the same number of chromosomes, but you can wind up with more or less than this actual number. Okay, so about 5%, that's 1 in 20 pregnancies wind up being aneuploid. Okay, the reason for aneuploidy, the reason for having the wrong number of chromosomes is because of non-disjunction, or the failure of homologous chromosomes or sister chromatids to separate during meiosis. So remember, this is meiosis, this is the formation of gametes, and this causes the sperm or eggs to have more or less than the normal number. Okay? So non-disjunction here we are looking at meiosis, remember, and this is normally what happens. Okay? You have these homologous chromosomes lining up on the metaphase plate, then they're split, then the sister chromatids are split again, leaving you with these haploid, leaving you with these haploid gametes at the end. What can happen, though, is that these pairs fail to separate. So if they fail to separate, you wind up with a non-disjunction here, and you wind up with the wrong number of chromosomes. So if you wind up with meiosis 1, all of your gametes have the wrong number of chromosomes. If you wind up with a non-disjunction in meiosis 2, it's just sister chromatids failing to separate, and so only half of your chromosomes are going to be aneuploid. Only half of your chromosomes are going to have the wrong number. So again, this is important to be thinking about meiosis, the meiotic process, and what would happen if those homologous chromosomes or if those sister chromatids failed to actually segregate into their individual gamete cells. So a very common form of this is Down syndrome, 0.1% of all births, and so it's not all that common. Most cases of Down syndrome are caused by trisomy 21, meaning you have an extra 21st chromosome. And these people usually look a little bit different, usually have a little bit of a lower IQ or are kind of shorter. Males who are Down syndrome are typically infertile. They live a shorter lifespan. And this is just an image of a person who has Down syndrome. And I don't know if you've ever met someone with Down syndrome. They're absolutely lovely people. They're really great to talk to and spend time with. So it's definitely not something that should be stigmatized. It's just something that happens. So trisomy 21 is the result of non-disjunction, usually during egg formation. It can also be due to issues in sperm formation, but it's usually due to eggs. And this frequency of non-disjunction, the amount that this kind of aneuploidy happens, increases with maternal age. So the older the mother gets when she has kids, the more frequently this is going to happen. So if you're 20 years old when you have a kid, your odds of having a child with Down syndrome are about 1 in 2,000, not particularly high. If you wait until you're 25, it goes to 1 in 1,200. If you wait till you're 30, it's 1 in 1,000-ish. If you wait till you're 35, it's 1 in 365. Sorry again. Which is still pretty good. But if you wait till you're 45, it becomes almost 1 in 32, which is staggering. That is a huge number compared to this 1 in 32 is huge compared to this 1 in 1925. So the older you wait, or the longer you wait to become a mom, the higher your odds of having a child with Down syndrome become. There's also Turner syndrome and Klinefelter's syndrome. So you've probably met people with these conditions before. This is kind of from the textbook. Usually have a lower than normal IQ and other phenotypes. That's not really true. I have friends that have Turner syndrome and they seem completely normal. They have normal jobs. They're no different than you or I, actually. And this is just a statistical thing. Maybe sometimes they have a lower than normal IQ. I've never met someone with Klinefelter, but apparently they are quite tall. But Turner syndrome, it's also not really true that they're sterile either. They just can't have, they just have problems having offspring. But this would be a female with an X and then a nothing. X naught it's sometimes called. And that's not a huge deal just because they already have that other X chromosome that they can use to compensate for it. Klinefelter syndrome are males who are XXY, so they have 2 X chromosomes and that Y chromosome to go along with it. These aren't things that I want you to know specifically. I'm just showing you some examples of aneuploidy. So you can use this thing. I referenced it in the lab this week, but you can use in vitro fertilization to screen embryos so that only embryos that do not have genetic conditions are actually being implanted. And there are some ethical issues that we talked about in class, although certainly less so than that of CRISPR. But should you screen embryos for things like height? Or should embryos be screened for things like hair color or gender? Or what should we be doing with these? Because you can tell those things. We can tell those things from genetics. And so I'm just leaving that as kind of an open question to you about whether or not we should be screening these embryos for anything other than like serious diseases. So this is the kind of the process of in vitro fertilization here. They're taking this big needle thing and injecting genetic material back into a mother. Actually, here I'm wrong. They're taking one of the cells and then they're going to subject this cell. This is a developing embryo here, and they're going to subject this one particular cell to genetic testing. And then if this passes the test, if it says, okay, this child does not have, for instance, Tay-Sachs disease or Huntington's, then they're going to take that the rest of this developing embryo and put it into the mother where it will finish its developmental cycle. Yeah, I'm just going to kind of gloss over that. So you can have other chromosomal abnormalities within the chromosome itself. Okay, you can have things like deletions happen. You can also have duplications happening. You can have transversions happening as well. So a deletion is sort of obvious, just a little piece of the chromosome is removed and deleted. A translocation would be taking a piece of the chromosome and moving it to somewhere else. And a transversion would be taking a piece of the chromosome, flipping it upside down, and just kind of putting it back. Okay, so an example of one of these would be krogh, which is a deletion on the short arm of chromosome 5. And here is a child who unfortunately has this genetic condition. You can see there are some potentially phenotypic abnormalities. And children who have this condition are usually severely developmentally disabled. And it's showing you here on chromosome 5, this is called a karyotype, by the way. It's just all of your chromosomes arranged. But this chromosome, I don't know if this is the maternal or paternal chromosome, but again, he got this one from his dad, this one from his mom, or whatever, however you want to think of it. And you can see chromosome 5, part of it has been accidentally deleted here. So he wound up with this unfortunate condition. When the broken pieces rejoin the same chromosomes in opposite locations, that is called a translocation. So again, translocation, they're just pieces of chromosomes are getting flipped on each other. And then if you have a error during recombination, you can wind up with a duplication, where a segment of the DNA is actually just getting copied more than once. So here's a deletion. You have A, B, C, D, E, F, and then on this other chromosome, you just have A, B, C, E, F. Part of it got deleted. You have inversions where pieces of the DNA are actually switching with one another on the chromosomes themselves. So you have A, B, C, D, E, F, and then you have A, B, C, E, D, F because they got switched. You have translocations where segments of the DNA are just switching parts where it isn't inappropriate. So maybe you have two chromosomes that are non-homologous, and you have UVWXYZ, and it's switching with this chromosome here. So you have A, B, C, D, Y, Z, and UVWXEF. Okay, so that would be a translocation, switching between non-homologous pairs. And then you can have duplication. So instead of A, B, C, D, E, F, you have A, B, C, D, D, E, F. And so here, just kind of a table that discusses all of the different things that we've been talking about. Here are these X-linked disorders. So those are just affecting your sex chromosomes. Okay, those are affecting men more often because we don't have another X chromosome to compensate. All we have is this puny little Y chromosome. Autosomal recessive disorders. So autosomal, not X-linked, not sex-linked. These are on the rest of your chromosomes. And in order to get one of these, you need to wind up with both copies of the allele. Now we have autosomal dominant disorders. Interestingly, polydactyly, so having 6 fingers is dominant. If you just wind up with one of those alleles, you wind up with more than 5 fingers or toes. We also talked about aberrations in chromosome number. So if you wind up with the wrong number of chromosomes, you wind up with some different conditions and aberrations in chromosome structure. Bad stuff happening to your chromosomes before they're actually getting to you. So that would be things like deletions, transversions, translocations, deletions, duplications, inversions, and what was the other one? I've forgotten it. Deletions, okay, translocations.